Thank you. Thank you for inviting me to talk here today. Um, First, I want to mention that I'm a molecular microbiologist, so my talk may be a little bit different, but I still hope you'll find it interesting. Um, in my lab at Center for Infection and Immunity at Columbia University, within the past 10 years or so, we've done a significant amount of work in respiratory disease surveillance. Now, in the last 6 years, we've done a lot of work with Enterovirus 68. And it began kind of by accident, which I'm going to mention, and it was at a time when really nobody else was studying this virus. So, obviously, as the name implies, Enterovirus 68 is part of this larger group of viruses, which we call enteroviruses. Now, this group includes viruses that we call rhinoviruses, polio, Coxsackie viruses. All of these are grouped into the genus Enterovirus. These are ubiquitous viruses. They're probably the most common RNA viruses infecting humans today. There are millions of cases, I should say millions of infections each year. The CDC estimates there's approximately 15 to 30 symptomatic infections each year and plenty more of asymptomatic infections. The virus is transmitted, or viruses, enteroviruses are transmitted through mostly fecal-oral route and respiratory droplets. And if a person does come down with a symptomatic infection, well, there's quite a huge range of symptoms that these viruses may cause, as I'm sure most of you already very well know. One of the things about enteroviruses, their neurological involvement is very well known. So I do want to briefly mention as to really go into detail as to what enteroviruses are genetically. I'm not going to make it too detailed, but enteroviruses, they are grouped taxonomically into the order Picornaviridae, family Picornaviridae, genus Enterovirus. Basically, they are part of this very large group of RNA viruses, which we call picornaviruses. Now, enteroviruses in particular, they're non-enveloped, meaning they don't have a lipid envelope outside of the virus capsid or virus coat. Their genome is a single strand of RNA encoded in positive sense. Their structure is icosahedron, and I have two pictures of enterovirus, sort of 3D structures. These are enterovirus C viruses, but they all basically look similar to this. And for viruses, they're rather small. They're only about 30 nanometers in diameter. Now, within this genus, enterovirus, there are currently 12 species. And these are named enterovirus A through enterovirus J, and rhinovirus A through C. Rhinoviruses, for the longest time, actually were part of their own genus. Now, with the advent of sequence-based taxonomy, it was found that the viruses actually are very similar to the rest of the enteroviruses, and now they've put them together in this one genus. I highlighted in yellow, and I'm sorry, the thing on the bottom kind of shifted up, but in yellow, the viruses that infect humans. So enteroviruses A through D are normally found in humans, and of course rhinoviruses A through C are found in humans. Now, these viruses are host-specific, so generally you only find them in their particular host. Sometimes they do switch hosts. For instance, in zoos you can find chimpanzees with human enteroviruses, and you can have people who have picked up other enteroviruses from chimpanzees. So they do kind of switch hosts, but in general they really are host-specific. Now, within each enterovirus species, there are a number of what we call serotypes. So we subdivide them into serotypes. Now, in the past, this was done by serology. So basically, these are variants that basically are not neutralized by sera from one particular serotype, but then they are not genetically different enough to be called a new species. And there's a lot of different serotypes of enterovirus. Currently, we're up to, I believe, 122. Now, these numbers are assigned sequentially, so whenever a new serotype of enterovirus is discovered, it gets given a number. Now, within species enterovirus D, there are currently 5 serotypes. 120, 111, and 94 have been discovered in Africa. They're extremely rare. Very little is known about them. And it's really the enterovirus 68 and 70 that are really of significance in human infection. Now, One thing I want to mention, a lot of people sometimes ask why Enterovirus 68 is called Enterovirus D68. And basically, this is just from taxonomy. It's an enterovirus. The reason we give this D letter, it's because it's species D. So it's enterovirus species D and the sequential number of serotype 68. The genome of enteroviruses is, as I mentioned, one single strand of RNA. Approximately 7 to 8 kilobases in length. For Enterovirus 68, it's about 7.3 kilobases, which is about average for RNA viruses. It contains a single long open reading frame, so basically all of this from here to here, that encodes one long polyprotein, which is then post-translationally cleaved into individual components that then make up the virus. Now, flanking this open reading frame are two regions which we called UTRs, or untranslated regions. One is found at the 5' end, one is found at the 3' end. The 5' end is important for several reasons. It is important for the virus biology because it contains something called an iris element, which is essential for initiation of translation. For diagnostics, however, it's very important because the vast majority of diagnostic assays target that particular region of the virus, the 5' untranslated region. This region is extremely conserved amongst enteroviruses, not only amongst different serotypes, but also amongst different species. So whenever someone sends out a test for enterovirus, very often the test comes back either positive or negative for enterovirus, but with no other information other than that, because PCR tests within the UTR region, while they're very sensitive, they cannot type or differentiate enterovirus serotypes. This is done by PCR and sequence analysis of other parts of the genome. So within the open reading frame, there are basically three regions. One is called P1, P2, and P3. The 2 and 3 basically called for non-structural components, which means viral enzymes like the polymerase, helicase, and various proteins that interact with the host of, during infection. The P1 region codes for the viral capsid, which is the viral coat. So basically this region right here. It contains 4 proteins, VP4, 2, 3, and 1. These come together to basically give the virus its shape. Now, VP4 is actually in the internal side of the capsid, so it's not surface exposed. It's the other 3 that are, and it's really this protein, VP1, that's the most external, the most variable, and the most immunodominant enterovirus proteins. So it contains most of the antigenic sites within enteroviruses, and therefore, in most cases, when one needs to identify the serotype present in a specimen, one would amplify by PCR this region, generally VP1, and obtain sequence, and then that would readily identify the serotype. Additionally, most serotype-specific assays, let's say if you want to, want to do an assay to identify whether Enterovirus 68 is present or not, would target the VP1 region. Only because this is by far the most unique region among each serotype, and you would find very little cross-reactivity with other serotypes. Okay, so a little bit about history of Enterovirus 68. There is, last year during this major outbreak, there was a big misconception in the press. A lot of people reported this virus as being new, which is obviously not true. It's not new at all. It was actually first isolated in 1962 in California. There was an outbreak, and this virus was isolated from 4 different kids diagnosed with pneumonia and bronchiolitis. Now, the studies analyzing this virus were published 5 years later. These 4 isolates were compared and were shown to be basically identical, but very different from any other viruses known at the time. So there was one particular isolate where most of the biological work was being done, and it was named Forman, which is what we call the reference strain or the type strain for Enterovirus 68. So all work thereafter was always compared to this type strain. Now, I do want to mention this strain basically doesn't exist in circulation anymore. It has changed so much that whatever comparisons we do are kind of irrelevant because this virus has changed so much, as I'm going to mention in a second. Subsequently, it was classified as an enterovirus. This Vermont strain was classified as an enterovirus, and it was actually the first one given a serotype number designation. So until then, all enteroviruses were either called polio, Coxsackie A, Coxsackie B, or echoviruses. And it was decided to have some kind of a common nomenclature from now on, and thereafter, starting with Enterovirus D68, all the other enteroviruses were named sequentially. Now, very little was done for quite a while, almost 30 years with this virus. One of the reasons was it was rare. Outside of this initial report in the 60s, there was really nothing in the 70s, 80s, 90s. There was some work done with a virus, an isolate, which was called corn, and was classified as rhinovirus 87. This was also isolated in the 60s. With, again, advent of molecular biology and comparative genetics, scientists actually compared the two strains and found them to be identical, meaning they were the same virus. So this corn isolate was actually misclassified as a rhinovirus, where it was in fact an Enterovirus 68. Now, why would such an error or misclassification take place? This is because Enterovirus 68 is biologically very similar to rhinoviruses. Genetically, it's more related to enteroviruses. However, it is acid sensitive. Majority of enteroviruses aren't. They need to pass through the stomach into the GI tract, which is the initial site of replication. Enterovirus 68 is acid sensitive, just like rhinoviruses. It grows better in tissue culture at 33 degrees Celsius, not 37, and that's the temperature where most enteroviruses grow better at. This is because it's associated with the upper respiratory tract. That's the temperature that it's sort of evolved to grow better at. And this is actually true. We found this in our lab, where we have had some isolates which grow very well at 33 degrees Celsius. They basically do not grow at 37, which is bizarre because there's only a 4-degree difference. They either don't grow or grow 10, 20-fold less. So this is another sort of similarity to rhinoviruses. And of course, up to that point, Enterovirus 68 was strictly associated with a disease of the respiratory tract similar to rhinoviruses. So why wasn't much more done with this virus? Again, it was very, very rare. How rare? This is a table from a morbidity and mortality weekly report from September 15th of 2006. This was data compiled from enterovirus surveillance between the years of 1970 and 2005. So here they basically compiled all the data, all the isolates of enteroviruses from isolated from various syndromes, and basically tried to identify which were the most common enteroviruses circulating in the United States within that time period. Now, if you look, I have, you can't maybe really see it, but I have an arrow pointed to Enterovirus 68, and as you can see, it was one of the most rarely reported enteroviruses during that span, with a total of only 26 reports within that long time span. The highest number of reports was 11 in 2003. In all cases but one, they were detected from respiratory specimens, mainly from children. However, there were a few adult cases as well. There was one case where this virus was identified by PCR from cerebrospinal fluid of a young male. Now, this was only reported in this report. It was not, to my knowledge, was not published anywhere else. Now, this study had several limitations which were acknowledged, one of them being the vast majority of this data was obtained from isolates of the virus, and while some enteroviruses are readily isolated, Enterovirus 68 is not the easiest virus to isolate in tissue culture. We found this out ourselves in our lab. I've spoken to other collaborators at New York State Department of Health, for instance, and a lot of viruses just don't grow in tissue culture, and we don't know why. So that's one bias. Additionally, there was a lot of enteroviruses which we call Coxsackie A viruses, a few particular serotypes which basically do not grow in tissue culture whatsoever. So obviously they were severely underrepresented. Nevertheless, this was a very good overview of the rarity of this virus in the United States up to that point. There was a report that came out in 2011 which documented another infection of Enterovirus 68 in the central nervous system. So there was that one initial report which I mentioned that was reported in 2005, and here was another one. This one was much more detailed, where it was a case in 2008 from a 5-year-old male where there was a respiratory disease going around in his class. He picked up whatever this disease was and very quickly progressed to neurological impairment, flaccid paralysis, pneumonia, and he died. Now, this was another case where post-mortem, they analyzed his CSF, and Enterovirus 68 was amplified from the CSF, linking this virus to the disease. Now, this is important because, as I'm sure a lot of you know, and as I'm going to mention, there's a lot of these flaccid paralysis cases associated with Enterovirus 68 now, and so far, in none of the cases has the virus been amplified or detected in the CSF. So this leads me to the work that we've done. So up to that point, as I mentioned, very little was known. It was a very rare virus. In our laboratory, in the past decade, and actually still, we do quite a bit of enterovirus surveillance. We collaborated with a lot of individuals from the US as well as different parts of the world, where we basically analyze samples looking for, well, detecting whatever's there, but also looking for novel pathogens. So we were very interested in pathogen discovery. We do this in a number of ways. The mainstay, of course, is PCR. We've used microarrays. We also now use high-throughput sequencing. But the most cost-effective way is PCR. However, there's quite a bit of respiratory pathogens out there. I mean, enterovirus, rhinovirus, flu, RSV, what have you. They're all the bacterial pathogens. It's not cost-effective, especially for a research lab, to do individual PCRs on all of these per each sample. What we have done is develop a number of multiplex assays where we basically screen for 10, 15, 20 agents in one shot. This cuts down the price, speed, and allows us to get a lot of information very, very quickly. We've developed these, what we call panels, and this assay is called mass tag PCR for respiratory agents, hemorrhagic, meningoencephalitis, tick-borne, enteric, what have you, and sort of it's still the mainstay of our surveillance strategy. So this is how we first came across Enterovirus 68. In May of 2009, we started a project with the New York City Department of Health. This was a pilot project to set up a respiratory surveillance program in New York City. Samples were collected throughout the city and sent to us usually weeks after the initial case. We weren't diagnosing anyone. This was sort of just to see what was circulating during the H1N1 outbreak. So samples began to be collected in May 2009, so very early in the H1N1 pandemic, and continued to be collected till May of 2010, so basically 13 months. These were nasal swabs collected, and samples were from children as well as from adults, and they were sent to us, and we analyzed them by multiplex PCR for 14 respiratory viruses. Now, what this table shows is the results of this study. We published this actually in 2011 in June. We looked at a total of 940 samples. We found a viral agent in about half, influenza A in a little bit over 20%. They were all H1N1. And then we found over 200 enterovirus positives. So this was, again, an assay that targeted the UTR region. We couldn't type what it was. We just knew whether it was an enterovirus present or not. So for each one of these samples, we then went back with an additional assay to try to identify the enterovirus, the serotype that was present. 185 of them were rhinoviruses, which was not unexpected, as rhinoviruses are extremely common. But what we did also find was we had a total of 24 non-rhinovirus enteroviruses. And there was this one particular spike in samples in September and October, where there was a total of 16 enterovirus positives, and all of them, when we typed them, were Enterovirus 68. They were all similar, the sequences were all similar or basically identical, which told us that these people were infected with the same strain of Enterovirus 68 that was circulating in New York City at the time. Now, why was this interesting? Well, because at that time, like I said, there was a total of maybe 26, 30 cases reported in the US over the past 30 years. Here we had like a localized outbreak, which we thought was unusual. Now, we wanted to pursue this a little bit further, mainly because the strain that we found, when we analyzed the sequence, was quite different from the strains that were previously deposited in GenBank and basically, around in the 60s, 70s, what have you. So we wanted to look at this a little bit further. Oops. Concurrently to this study, we were doing quite a bit of other surveillance studies with other collaborators. We were doing a study with collaborators from the Gambia, where we had 816 samples that were collected over a calendar year. And also, we were investigating an outbreak in Senegal of 41 samples collected over 2 months. South Africa, over 1,000 samples collected over 2 years. The New York City samples I already mentioned. And Arizona, where we looked at the incidence of H1N1 in Navajo children. So these were over 600 samples collected over 1 calendar year. The majority of these sample sets were pediatric cases, so it was kind of biased from that aspect. But what we wanted to see is whether the enteroviruses that were present in these specimens that we found, whether we could detect this previously very, very rare virus. And sure enough, in every single sample set, we did, albeit, again, very rare. It was a very rare virus. In the Gambia, we found it in 5 samples out of 816. They were all clustered within 1 month, so there was probably a localized outbreak then. In Senegal, out of the 41 samples, the vast majority had a rhinovirus, but we did find 3 Enterovirus 68 positives, and they were actually very similar to the sequences we found in New York City. In South Africa, in samples from 2000 and 2001, we found 8 cases interspersed. These were all from hospitalized children with pneumonia. In the US, we found, from Arizona, we found only a single case out of the 600. But what we did achieve here is show, okay, this virus is, yes, it is rare, but if you actually look for it, you'll find it. Not only that, when we looked at the genetic sequences of these viruses, they were very different from each other and from the viruses that we knew up to that point. So as we were preparing a manuscript to show this, 2 months after our initial report from New York City, basically an avalanche of reports from all over the world started popping up with reporting Enterovirus 68 outbreaks. It began in Europe, in the Netherlands, followed by Asia, in Japan, several reports from Japan. There was one in the Philippines, in Thailand, as well as a report from the CDC, basically reporting several clusters in the US and a cluster among American Indian children. Now, these are reports only from fall 2011 up to, I believe, April 2012. So it's just within that time period, there was basically quite a bit of paper showing these spikes, these sort of upsurges in Enterovirus 68 cases. Most of these were in pediatric sample sets. So this was surveillance studies where people looked at viruses that were present in pediatric cases, and lo and behold, they started finding a lot of Enterovirus 68s. And in Europe, especially in the Netherlands, they found not only that a lot of samples, that they found that each year they seem to have been increasing. They started looking back earlier on in the decade, and they found very few. And as they kept going closer and closer and closer, there was an upsurge in cases. Well, another thing that a lot of these reports provided us is genetic sequence of these viruses. So in many cases, the scientists working on these outbreaks, they amplified partial genomes of the virus, deposited the sequence, and in combination with the sequences that we had from Africa, from the US, we basically combined this data to see what's out there right now. What is this virus? How is it different from the viruses that was circulated, that was around 30 years ago? So this is a phylogenetic tree, which basically shows the evolution of this virus up to 2010. So we published this in Journal of General Virology in 2012. What you can see from here, that first of all, between 1960 and really like mid-90s, there was really nothing there, mainly because this virus was rare. There was really no sequences deposited. Only in the late 90s, more cases began to be seen, and really in the mid-2000s, where a lot of new cases and a lot of new sequences were deposited and allowed us to sort of analyze this a little bit better. But one thing that kind of comes across very clear from this is that this virus evolved or split into three very distinct lineages, which we called clades, clade A, B, and C. At the time, a good proportion of the outbreaks that were going on in 2009 and 2010 were from this particular clade, clade A. There were not a lot of sequences from clade B. However, all of these, and I have, so all of these basically branches represent a single sequence, and I have them color-coded to represent which country they came from. And there was an outbreak in the Philippines where 24 children were diagnosed with Enterovirus 68, of which 2 died. So most of them were in ICU, and 2 of them died. There was also a fatal case in Japan as well. But at the time, most of them were from clade A, and then again, we sort of differentiated them into 2 other clades. But another thing became clear, that not only has this virus split into these 3 clades, they were all circulating globally. And in the Netherlands, for instance, all 3 were circulating within the same year. Now, why is this important? Well, because if you're infected with one, you can be infected with another clade and another clade. So just because you're infected with one will not protect you indefinitely. What I have shown here on top is an amino acid sequence alignment, basically of VP1 region. So it's VP1 protein. So it's the most variable protein of enteroviruses, and the mutations that we found in these clades, clade A, B, and C, relative to this type strain, were all found in regions of proteins which are thought to be associated with receptor binding and immune system, in places where basically the virus comes in contact with the immune system, in basically antigenic sites, which led to our hypothesis that basically these mutations allowed this virus to evolve and basically sort of evade the immune response as it kept evolving over many, many years. There were also changes within a region which we call the hypervariable region within the UTR. So the UTR, as I mentioned, is a very conserved region of enteroviruses. There is, however, a stretch of about 100 nucleotides in enteroviruses which are variable, meaning they tolerate mutations very easily, and we actually don't know what it does. Here, clade A, all members of clade A developed a deletion. Basically, we're missing 24 nucleotides, while clades B and C were missing an additional 11 nucleotides. What it means, we don't know, but it may have allowed the virus to be more efficiently translated, basically may have enhanced its biology, which yet again may have led to aided its expansion. After our reports, we've done some other work with other collaborators where we look for this virus in other places. We have some unpublished work with collaborators from Peru, for instance. But recently we published a paper where we looked at analysis of respiratory disease clusters in Cambodia. So in Cambodia, there's a very high rate of undiagnosed acute respiratory infections. So this work was done with Naval Research Institute 2, where they collect samples from several provinces in Cambodia and analyze them for influenza, dengue, and malaria. They identified 6 clusters, and by cluster I mean a set of samples collected within kind of a defined time frame where most of the individuals show the same symptoms and the same geographical area, of course. 6 of these clusters were sent to us for analysis. They had a total of 85 patients, and we looked for viruses by a multiplex PCR to see whether we can find any pattern. Three of these clusters were from the dry season, 3 were from the rainy season in Cambodia. So as some of you may know, those basically are the 2 seasons that they have there. In the dry season, we really didn't find anything, unfortunately. One thing that was actually very interesting is for all these cases, we had a very well-defined clinical data for all the individuals, not only for the clusters, but individuals within the clusters themselves. And in the 3 rainy clusters, they were called KDO2, KSO2, and RKO1, basically based on the geography. In one of the clusters, there were 20 cases over an 8-day period collected in August of 2009. We identified Enterovirus 68 in 6 of these. Now, when we analyzed the sequence, they were all from this clade A, which was very similar to the clades that we found in New York City. They were all adults, just like the cases that we found in New York City, interestingly enough. 5 of them presented with pharyngitis, headache, cough, and malaise, and 1 of them presented with abdominal cramps and joint pain, which were atypical of an enterovirus infection, which we think probably may have been due to some other pathogen. In another cluster of 22 cases over a 7-day period, this was a mixture of adults and kids. In most of the kids, we found RSVB, but in one of the adults, we found another case of Enterovirus 68, very similar in sequence to these. And this is actually a phylogenetic tree which shows where these new strains that we found fall in, so that they're actually shown with this asterisk. And in the 3rd cluster, these were all cases of children under 5 years of age, a total of 11. They were all diagnosed with pneumonia. We found RSVA in 9 of them. 1 of the kids was co-infected with Enterovirus 68 as well, and this one was part of this clade B, which initially, in our analysis from 3 years back, it was fairly rare. Well, as time went by, there have been more and more reports of clade B circulating in other parts of the world. Here, I just listed 4 reports between 2013 and 14. There's actually more than these, but these are sort of the more interesting ones, about Enterovirus 68 in Kenya, where all 3 clades were circulating, in Italy, clade A and clade B, and then New Zealand and France. And this all culminated with what happened last year. So in mid-August, there was a huge upsurge of severe respiratory disease cases reported in basically in Midwest and Missouri, and in Illinois as well. This was reported first by the Morbidity and Mortality Weekly Report at the beginning of September. Now, the data that I have here, this text, it's mostly data summarized from the CDC, that by mid-January, there was a total of 1,153 EVD68 confirmed cases in 49 states and District of Columbia. Now, another interesting tidbit about this is in this year, in 2014, the CDC received 2,600 specimens for enterovirus lab testing, which is much more than they usually get. Of these, 36 were positive for Enterovirus 68, and only 33 were positive for rhinovirus or some other enterovirus, which is extremely bizarre. In all the work that we've done and published on respiratory disease, and what others have published, rhinovirus is usually the predominant virus that we find in nasal swabs. And if it's not the predominant one, it's usually the second most predominant one. Here, Enterovirus 68 was by far the most predominant one that year. There was also a total of 14 patients that died and had samples, respiratory samples submitted for postmortem testing, and Enterovirus 68 was detected in these patients. Now, soon after the outbreak was reported, the CDC sequenced the virus, several isolates that were circulating at the time, and they were part of this clade, which we called clade B. I have listed in stars as basically the new isolates. This is a tree that I made, it's kind of like a simplified tree to show where these new viruses fall in. There were other isolates that kind of fall in outside of clade B. There may have been actually a new clade that has evolved called clade B, but the vast majority of them were within this particular clade. In addition, as I'm sure a lot of you know, there was, coinciding with this outbreak, there was an increase in reports of acute flaccid paralysis in children, first in California, but then in Colorado as well. CDC issued an alert for this particular syndrome. What the causative agent was unknown. However, it coincided with the outbreak, and a lot of the individuals who suffered from this syndrome had a respiratory disease within 2 weeks prior to the onset of symptoms. And additionally, in several of them, Enterovirus 68 was amplified from nasal secretions, linking potentially this virus as to the cause of this new syndrome, which is now called acute flaccid myelitis. Recently, several papers have come out in Lancet, kind of more directly linking Enterovirus 68 infection in this syndrome. More specifically, a paper that just came out, I believe at the beginning of the month, from a lab of Charles Chu, where they actually looked at samples going back to 2012 and tried to link Enterovirus 68 with this syndrome. So what they did is they had a total of 24 patients, I believe, with this syndrome, and looked by PCR as well as high-throughput sequencing to see whether they could identify Enterovirus 68 in their nasal secretions, blood, as well as the CSF. In half of the patients, they did detect Enterovirus 68. In one patient, in one patient's blood, they identified Enterovirus 68. However, in none of them were they able to identify the virus in the CSF. This study sort of added to the association of this virus to flaccid myelitis. However, it's still the smoking gun was not found. So there's still plenty more work that needs to be done to actually linking this syndrome to Enterovirus 68 infections. So what's next? We know a lot about epidemiology of Enterovirus 68. So it's here to stay, obviously. There's probably going to be new clades that will arise. This virus is mutating fairly rapidly. Now, we do know a lot about its epidemiology. We know very little about its biology, I should say, virology. There's been some work done regarding receptor association, but for instance, they found that this virus tends to bind to sugars in the upper respiratory tract, but it doesn't bind to sugars, which I thought to be the receptors, of the lower respiratory tract. However, that does not explain why it can cause infections of the lower respiratory tract. So obviously, it uses some kind of other mechanism to do so. There is very little known about whether clade A or clade B or clade C is more pathogenic or more easily transmissible than the other. For instance, clade B, which was the source of the outbreak last year, is it more neurotropic? Well, in this latest paper by Charles Chu, they identified several mutations present in this clade B strain, which they associate with enhanced neuroinvasiveness because they found similar mutations or similar, I should say, amino acids in polio and I believe Enterovirus 71, which is also neurotropic. And of course, therapeutics right now, it's just supportive treatment, so that's still ongoing. There's one last thing I want to mention, and this actually has nothing to do with Enterovirus 68. We have done, as I mentioned, quite a bit of work with respiratory viruses. We analyzed all these different sample sets, and you look at all these different sample sets, you're going to find something interesting. And we did. In 2013, we published 2 papers. Interestingly enough, they both begin with the title Genomic Analysis, which was kind of an accident. But we identified novel serotypes of enteroviruses, which are now called Enteroviruses 118, 105, and we also did some work with Enterovirus 104. What's unusual about these is, they are all part of Enterovirus C species, but they all seem to be respiratory tract tropic and cause mild respiratory disease. So this is a phylogenetic tree which shows all Enterovirus C serotypes. There's sort of like two different clusters. Here's one which has polio and a lot of Coxsackie viruses, and then there's this group. These three cause flaccid paralysis, and then these guys, all of them, have only up to now been identified in respiratory samples. They have not been identified in any fecal samples or anything like that, and they are all associated with respiratory disease. Now, why is this interesting? Well, because these are thought to be rare. Enterovirus 68 was initially it was thought to be very rare. Additionally, these viruses seem to be evolving from each other. So the ancestral strain to these was Enterovirus 104, which actually was discovered by a Swedish group in a Swiss group in 2008. The serotypes that have come after, like 118, 105, have mutated in certain places, and the mutations that we see are very similar to the mutations that Enterovirus 68 has acquired from its initial ancestral strain. So whether these become more prevalent in the future, we don't know, but it is something to think about because there's not just Enterovirus 68, there's plenty of other enteroviruses that can potentially in the future cause outbreaks, and we're really not thinking about it yet. And that's it. I just want to acknowledge the people in my lab, mainly Dr. Ian Lipkin and all the people, all the collaborators that helped me with this. Thank you.